Oops, that didn't all come through on the screen. Oh, there it is. It's animated. The Illumina representatives came to visit us a couple of weeks ago. Illumina is the company that makes the current state-of-the-art instrumentation. And I took the slide. I didn't realize it was animated. So they said at their visit to us at Mayo that they're going to be, by the end of this spring, they're going to have a performance of 1.14 terabytes per run. Terabases per run. Or basically 10 times 10 to the 12th bases per run. And that's about 2 weeks if you count data sample preparation and all. And the bottom line is that that instrument can do 12 people per run for about $20,000. So I'm just let that sink in for a minute, and we're accelerating downward. Next slide. How is that affecting the world economy? This was in Nature, a science journal, prominent science journal, last October. There are these bars, silos, are the amount of next generation sequencing devices in individual countries. We have in the U.S. about 1,000 of them. China at this time has about 150. These people here, there's a project going on, the 1,000 Genomes Project, which is basically to completely sequence 1,000 genomes. In the U.S., this is 2010 numbers. The U.S. will have done about 150 people at the end of last year. Asia would have done about 50. By the end of this year, based upon the capacity of these instruments, we will have done about 2,000 individuals. And in China, they will have done 15,000 people by the end of this year. And this is going to only accelerate. So I bet it'll be 10 times more than that by 2012. Maybe even more. So basically, the ability to get genomics information is easy now. And the price is going down. Next slide. This is a paper that was in Science Translational Medicine, another prominent journal, just a couple of weeks ago, that relates to what Karen and I do professionally. You don't have to sequence human genomes. You can actually target specific genes. And so this came from the National Institutes of Health and a scientist who's now at the University of Kansas. And they basically designed a way to sequence all the genes known in severe recessive childhood disorders. 448 of them. Things like Tay-Sachs, Hurler's, Hunter's, or diseases that result in early childhood mortality. The kind of diseases that prenatal screening is done so that those individuals can be those pregnancies can be not terminated or not allowed to continue. So this is a screening, prenatal screening or carrier screening for couples for almost all known major disorders. And this can be done in 8-fold multiplex for about $100. The group at Kansas is now offering it through their pediatrics hospital. And the interesting thing about this is that they found, so we can come back to this, they screened 100 people, and all those 100 people, everybody, all of them, on average, carried 3 of these mutations. That means that all of you in this room, if you were sequenced by this device, would be found to carry 1 copy of a recessive mutation that can cause severe, neonatal or newborn or young child mortality. And that doesn't happen very much because you have to marry somebody else who's a carrier. But that just means you all of us have these, and some of us have more, and most of us have at least one. The most important part of the paper. What does that mean, just to move into ethics just a wee bit, what does that mean for insurance coverage if everybody knows if we're all carrying something, right? That's a pre-existing condition, so let that sink in. Next slide. I'm doing this in my research lab. This was actually Vern said in a sermon a couple of weeks ago. I invited him down to Man Hall to hear my talk on the genetics I do in gliomas. And we are doing next-gen sequencing on 2,000 people. We're not doing 2,000 individuals' whole genomes. We're doing about 650 kilobases on those 2,000 people using this next generation sequencing in an effort to find the genes that predispose to glioma. This is within the range of something that I can do in my laboratory. We're actually doing it in conjunction with another lab at UCSF, and we used money from the recovery grant. I got one of the recovery grants. And it's going to cost, at the time we ordered this from the company, it was going to cost us about $200,000 to do this one experiment. I don't know what it would be now, but it would be far less than that. But of course, we're paying the price that we had to contract for. So one of the challenges for people like me, professional geneticists, is when do you jump in to do the experiment? Because the price keeps coming down. And we can write a grant, and the grant's not going to be funded for a year. In the meantime, I showed you the curve. Price is coming down, the capability is going up. This data, we'll have this data probably, well, it's supposed to be done on February 1st. Well, probably will be February 15th, and then we'll begin mining it. It used to be that mining the data was a challenge. But Illumina has now figured out that they can actually do the data annotation on a whole genome for $75 by shoving it into the cloud. Okay? So that it's really the processing capacity. The algorithms have been built.
What's a cloud?
The cloud is this, Annette can maybe describe it better than me. The cloud is this sort of thing up there that's basically a huge computational network. So when you do Wi-Fi, you're jumping it into the cloud. It's a big computational network. And they use this network computers to do the computations. I think that's what it is. Somebody else can correct me if I'm wrong. Next slide. Eat, and your eyes shall be opened, and ye shall be called geneticists. All right, so let's go to the statement. The 5 directions in the development of genetic technology. So this is just their list of the things that they think this document should be addressing or should be applied to. We could spend a year going through each one of these. All right, so we're going to just do a few of them, but I'll read these so you can get an idea of what it's talking about. Genetic engineering in agriculture. Biotechnology, including practices such as genetically engineering seeds or cloning plants and animals, and farming with the PH. Farming means using animals and plants to make drugs as little factories, walking, breathing, respirating factories. Molecular medicine, including practices involving stem cell research, genetic therapy, personal genomics, and SNP mapping. I do personal genomics and SNP mapping, as well as efforts to extend the longevity of human life to as much as 3 times today's average. Procreative activities, including prenatal testing and screening, genetically aided assisted reproductive technologies, ART, pre-implantation genetic diagnosis, PGD, and artificial creation of new life forms, synthetic biology. It's out there. We don't have an example of that, but if you have a question, then I can tell you how that works. Commercial delivery, including DNA testing for employment and health insurance. Remember, all of you are carriers of at least 3 severe recessive disorders. Trade policies, patenting of genetic material, and research processes. Social use or implications such as criminology and DNA evidence, potential implications including discrimination based upon genetic profiling, the practice of eugenics, and belief in genetic determinism. The next slide shows you what's happening with life elongation. This was just published in January. I know the first author, the last author, the senior author, Ron DePino. He's a professor of brain tumor biology and genetics at Harvard. Telomerase reactivation reverses tissue degeneration in aged telomerase deficient mice. I heard about this as I was running on the treadmill at the YMCA. Ron DePino was being interviewed on Science Friday. All right. Then, 2 weeks ago, Sue and I watched Stephen Colbert and Jon Stewart, and we were TiVoing it, and we were watching it, and it says Ron DePino. So Ron DePino is on Colbert. He said in this paper, in conclusion, this unprecedented reversal of age-related decline in the central nervous system and other organs vital to adult mammalian health justify exploration of telomerase rejuvenation strategies for the age-associated diseases, particularly those who are driven by accumulating genotoxic stress. There are drug companies working on reactivating telomerase. It's an enzyme that's shut down as we age, and is basically one of the main reasons that we age. And if you could reactivate the enzyme, one could potentially link the person's life. And this group did this in a mouse. In these mice, they didn't have Alzheimer's disease, but they had some neurodegenerative things. They were calling those symptoms, and those symptoms got better. You can't read this. I won't read it. But this means you can find this anywhere. Karen and I just went on the web just for briefly. I mean, you can find stuff all the time. Extinct woolly mammoth may be resurrected by scientists. So talk about synthetic biology. This is not really synthetic biology, but basically a Russian group has excavated a mammoth from the permafrost in Siberia. They transferred the carcass to Japan, and Japanese scientists are trying to extract the nucleus from that frozen material and put that nucleus into a fertilized ovum that has been denucleated, taking the nucleus out of an elephant, and then putting that renucleated cell into a maternal mother to give birth to a woolly mammoth. I'll let that sink in for a minute. I think it's going to happen. All right. Next slide. So now we're going to do a couple of some scenarios. We'll read them and then come through some of the things that this document said, and then we'll have a discussion. And Karen's going to do some talking about some of the moral parts of this document. So Frank and Jody are farmers in southeastern Minnesota. The farm has been in their family for 100 years. Every year they save back some of their soybean and corn seeds. The seeds are cleaned by an itinerant seed cleaner, Max. Max and his father have cleaned seed for Frank and Jody's family for many years. Any of those of you who are in a farm family or raised in a farm family know that you didn't have to buy seed from the seed company. You could hire a seed cleaner to clean a portion of your seed you held back for next year. Several the farms around Frank and Jody have been planting GM soybeans, genetically modified soybeans and corn that are resistant to Roundup. These farmers buy their seeds and herbicide from a large seed company. As again, those of you who know about this know that that is a contract you sign. You sign to buy the seed and you sign to buy the herbicide. It's all one big contract. 10 years ago, this did not exist. My cousin is a seed person in Southern Illinois, works for Farm Service, and this did not exist 10 years ago. The seed company has sent representatives to randomly sample Frank and Jody's soybeans. They find evidence that some of the grain is actually genetically modified. How did that happen? Well, it turns out there's pollen, right? And the pollen gets up in the air and can pollinize neighboring fields. The company's lawyers asked Frank and Jody to pay the royalties that the neighboring farmers have paid to use the seed. They are also told that they must not use Max's seed cleaning process. Frank and Jody have never bought GM seed. What should they do? Think about that. We'll go on. Possible outcome of the Human Genome Project. No more genetically imperfect people. Like Stephen Hawking. No more depressed people. Like Vincent van Gogh. No more physically challenged people. Like FDR. Well, no more blind people. Like Stevie Wonder. Well, no more people with multiple handicaps. Like Helen Keller. Well, maybe there's a lesson here. Like there's no two sides to every coin. This is about making perfect people. Next slide.